Good afternoon. I'm Dr. Brian Shapiro, a scientific content specialist at ATCC. Thank you for joining us for the latest installment in the ATCC Excellence in Research webinar series entitled Understanding COVID-19, a Global Pandemic, presented by Ms. Brittany Tang. Ms. Tang is a microbiologist at ATCC. In this webinar, Ms. Tang will give an overview of the novel coronavirus SARS-CoV-2. She will then explain the advancements in scientific and clinical research and how ATCC is contributing to the fight against COVID-19. If you have a question for our speaker, please use the chat function available through the webinar program. All questions will be answered as time allows at the end of the presentation. The recorded webinar presentation will be archived on the ATCC website, www.atcc.org. So with that, I'd like to welcome Brittany Tang.
Thank you, Brian, and thank you to everyone for joining our webinar today. Before we delve into COVID-19 and SARS-CoV-2, here is some background on the American Type Culture Collection, or ATCC. ATCC is a private nonprofit organization founded in 1925. The company is headquartered in Manassas, Virginia, but also has an R&D facility in Gaithersburg, Maryland. ATCC is a leading global supplier of high-quality biological materials, providing the scientific community with a diverse range of industry-standard products ranging from cell lines, microorganisms, to media and reagents. And of course, the focus of today's talk, SARS-CoV-2 related materials. In today's webinar, I will be going over the coronavirus disease 2019, or COVID-19, and its causative agent, SARS-CoV-2. I will discuss the nature and the background of the disease, characteristics of the virus, and advances in the field. To address the urgent need for diagnostics and interventions, I will provide an overview of current testing methods and the progress in the development of antivirals and vaccines. As I touch on the development and validation of diagnostics, I will also show how ATCC is supporting scientific advancements during this pandemic. There are 5 main areas of scientific research that will be covered: understanding the disease, understanding the virus, diagnostics, development of prophylactics, and development of therapeutics, as it is important to understand the complexity of the disease and the pathogenesis of its infectious agent to move forward with diagnostics for detection and surveillance, and the development of vaccines and antivirals. First, I will go over the characteristics of COVID-19, such as the prevalence, epidemiology, and clinical manifestations. Within these past few months, the respiratory syndrome caused by the virus SARS-CoV-2 has become a critical public health threat. The infection was first identified in Wuhan, China in late December 2019, where it was associated with a large seafood and animal market. Within the first month, the disease has spread to 23 countries, and confirmed cases jumped from 41 to over 9,000. SARS-CoV-2 spreads easily person to person through respiratory fluids via either symptomatic or asymptomatic patients. This rapid spread has caused significant impacts on healthcare systems and global societal disruption. Globally, COVID-19 has affected 215 countries, and there have been more than 6.9 million cases and 400,000 deaths and counting reported by the World Health Organization. Based on currently available epidemiological information, people of any age can be affected by COVID-19. However, it is likely that older adults are at higher risk for severe illness. As shown in this table, the rate of hospitalizations by age increases significantly in adults older than 65 when compared to other age groups. In addition to age, underlying health conditions are also a risk factor for severe illness. Medical conditions such as chronic lung disease, moderate to severe asthma, heart conditions, and liver disease increase the risk of hospitalization. As shown with data from the CDC, hypertension, obesity, metabolic, and cardiovascular disease were the largest risk factors. As its name states, the severe acute respiratory syndrome coronavirus 2 is a syndrome caused by infection of the respiratory tract. The infection results in severe inflammation of the lungs alveoli, which are responsible for gas exchange during respiration. In severe cases, infections are systemic, and the virus invades any tissues which express the ACE2 receptor in which it binds to. When septic, patients shed the virus through cerebrospinal fluid, urine, and feces, which exacerbates its ability to spread. Damage also occurs to the blood vessels, causing blood clots and microischemic strokes or pulmonary embolism. Symptoms typically appear 2 to 14 days after exposure, and illness may present with flu-like symptoms such as fever, cough, headache, sore throat, and chills. Characteristic symptoms of COVID-19 include shortness of breath, difficulty breathing, muscle pain, and temporary loss of taste or smell. Infection can transition from the milder flu-like symptoms to more severe pathology like viral pneumonia, pulmonary phase, or hyperinflammation syndrome, which can progress to acute respiratory distress syndrome, or ARDS. ARDS causes hypoxia, pulmonary edema, and infiltration of inflammatory immune cells into lung tissue. If unresolved, ARDS can progress to end-stage lung disease leading to death, which contributes to the unusually high mortality rate for COVID-19. With all of this in mind, understanding the nature, progression, and manifestations of COVID-19 help to define the characteristics of infection. But identifying the mechanisms used by the viral agent SARS-CoV-2 is crucial for developing diagnostics and treatment, which we will now delve into this portion of the webinar. Although much is still unknown, new papers, scientific research, and clinical advances are being made at an unprecedented rate. Luckily, our extensive history with coronaviruses can help fill in those gaps. SARS-CoV-2 belongs to the Coronaviridae family, which are large enveloped RNA viruses. Coronaviruses are distributed widely among mammals and birds and are of medical and veterinary importance. They can cause respiratory and enteric infections, but may also produce hepatitis or neurological symptoms, among many others. These viruses have also been identified in many species, such as swine, birds, cattle, and even fish, as well as humans. In humans, common coronaviruses include two types of alpha coronaviruses, 229E and NL63, and two types of beta coronaviruses, OC43 and HKU1, which circulates in humans and cause a common cold. During the past 2 decades, 4 large-scale epidemics have been caused by coronaviruses: severe acute respiratory syndrome 1, Middle East respiratory syndrome, swine acute diarrhea syndrome, as well as the current virus, SARS-CoV-2. SARS and MERS emerged respectively in 2003 and 2012, causing worldwide pandemics, while SARS struck the swine industry in 2017. All of these viruses share common characteristics by being highly pathogenic to humans or livestock and originating from bats. Most likely, future SARS or MERS-like coronavirus outbreaks will share these similarities in origin and host range. And because these viruses and the diseases they cause are closely related, the virus causing COVID-19 thus has been named Severe Acute Respiratory Syndrome Coronavirus 2. Genome-wise, coronaviruses are envelope-positive sets RNA viruses, mainly characterized by 3 unique features. 1. Club-like spikes on their surface. 2. An unusually large RNA genome. And 3. A unique replication strategy. The club-like spike proteins are used to bind to the ACE2 receptor in human cells during infection, while the genome is more than 30 kilobases long, is positive sense, single-stranded, and contains multiple open reading frames, or ORFs. The genome comprises of a leader sequence, ORF1A, which encodes proteins for RNA replication, ORF1B, which encodes genes for non-structural proteins that have a critical role in RNA synthesis, and also structural proteins required for virion assembly. Another interesting feature of this virus is its unique replication strategy, as it involves 2 RDRPs, or RNA-dependent RNA polymerases. The first RNA polymerase is primer-dependent non-structural protein NSP12, and the second is NSP8, which has a primerase capacity for de novo replication and initiation without primers, and is also important in transcription. Because replication and transcription of the virus depends on the RDRP proteins, these proteins have been tested to be effective targets for antiviral drug interventions. Several RDRP drugs, such as remdesivir, are currently being tested in clinical trials. SARS-CoV-2 is part of the Coronaviridae family, whose members are named after their crown-like appearance caused by their surface glycoproteins. The family includes the two subfamilies Letovirinae and Orthocoronavirinae. Orthocoronavirinae is subdivided into 4 genera, which includes alpha, beta, gamma, and delta coronaviruses. Alpha and beta coronaviruses typically infect only mammals, whereas gamma and delta coronaviruses infect avian species and sometimes mammals. Based on genomics and phylogenetic studies, SARS-CoV-2 is classified as a beta coronavirus, which also includes some of the more pathogenic viruses to humans, including SARS-CoV-1 and MERS. SARS-CoV-2 has also been found to be closely related to bat-associated SARS coronaviruses. Although the host reservoir of SARS-CoV-2 is likely to be bats, there are possibilities of intermediate hosts who may have become infected through saliva, feces, and urine of bats. Potential species identified with genomic and geographical studies include bamboo rats, pangolins, snakes, and possibly others. This map reflects the average temperature data from March to April in 2019 and was developed to predict the areas that are at risk for community transmission of COVID-19. Zones at the highest risk are within the green bands, in which colder and drier seasons are suggested to positively impact the strength of viral transmission, while zones at low risk are within the red bands, in which warmer seasons can negatively impact viral transmission by slowing it down. This as of which shows that seasonality may possibly influence the strength of viral transmission of SARS-CoV-2, especially given the consistent seasonal variation of the 4 endemic coronaviruses 229E, HKU1, NL63, and OC43. SARS-CoV-2 may be affected by climate, humidity, and presence of UV light. When it comes to viral infection, SARS-CoV-2 has been shown to be able to infect cell types from different species. This research paper, Functional Assessment of Cell Entry and Receptor Usage for SARS-CoV-2 and Other Lineage B beta coronaviruses by Letko et al., has demonstrated this by developing a synthetic biology approach to rapidly screen lineage B beta coronaviruses, which include SARS-CoV-2, for receptor usage and their ability to infect 5 different cell types. 3 of the 5 cell types used in the research, which are human lung epithelial cells, hamster kidney cells, and human colon epithelial cells, are ATCC items, which emphasizes the demand and contribution of our materials and scientific research towards understanding the biology of SARS-CoV-2, because credible materials lead to incredible research. To bring the focus back on the receptor usage and infectability of SARS-CoV-2, Letko et al. Demonstrated that the virus is able to infect these various cell types along with others by interacting with the host cell receptor ACE2 through the receptor binding domain in the spike protein. Interestingly, within the background of the SARS-CoV-2 spike protein, cell entry is very similar to the other clade 1 spikes tested, including SARS-CoV-1. These findings suggest that SARS-CoV-2 is capable of using the human receptor ACE2 as efficiently, if not more, which may help to explain the human-to-human transmissibility of this virus. Now that we have a better understanding of the disease and the mechanisms of the virus, we can talk about the types of diagnostic testing available and the importance of assay validation for viral detection and surveillance. To respond effectively to the COVID-19 outbreak, diagnostic testing is critical for tracking, understanding epidemiology, informing case management, and to suppress transmission. At present, diagnostic tests are categorized into 2 types: molecular-based assays and immunoassays. The most common testing methods being used in healthcare systems are RT-PCR, antibody-based tests such as ELISAs and lateral flow immunoassays, and rapid antigen tests. Molecular assays detect viral RNA to confirm infection, while immunoassays measure antibodies or antigens made in response to infection. The variation in testing effectiveness can be seen in this chart. Within 2 weeks of exposure, tests are unlikely to detect the presence of the virus, and symptoms usually take 2 to 14 days to appear. Once symptoms do appear, viral RNA can be detected by PCR, although the viral burden may be low. PCR using nasal swabs are more likely to detect viral RNA early, as the virus replicates in the nasal cavity and airways before migrating down the respiratory tract. However, PCR testing is less likely to detect RNA near the end of infection when the virus is being cleared by the immune system, as it typically takes 1 to 2 weeks for a patient to develop antibodies. During this time, antibody-based testing measuring the immunoglobulins IgM and IgG is beneficial to confirm what proportion of the population has been infected. IgM is usually the first antibody produced by the immune system and indicates that a patient may have been infected and their immune system has started responding, whereas IgG antibodies develop within 7 to 10 days after symptoms begin and remain in the blood after infection has passed, indicating a past event of infection and a potential development of immunity. Because of this delay, antibody-based tests are not recommended for diagnosis of acute cases. Only nucleic acid or antigen-based tests are verified by the CDC. The majority of these PCR-based assays target the genes ORF1ab, RNA-dependent RNA polymerase, envelope, nucleocapsid, and spike regions. On the immunological side, currently authorized assays measure IgM, IgG antibodies, total antibodies, or the spike protein. Usually, diagnostic tests and medical devices are subject to 510 approval from the FDA. But due to the urgent need for testing, current diagnostic tests are approved under the FDA Emergency Use Authorization Guidelines, which are lower bearing so that they can be approved and used much more quickly. However, further validation is expected. Currently, there are many in-house and commercial molecular and serological assays that detect the virus, which have been developed or are currently under development. Collated lists can be found on a variety of resources, such as the ones provided. In assay development, validation is crucial to ensure that assays are sensitive and specific to SARS-CoV-2. FDA guidelines are used to make sure tests are not cross-reactive to other organisms, and if so, there is a limit of detection to establish the sensitivity of the test to minimize false results. From assay development to validation, ATCC provides a variety of materials that can be used for various research purposes. The regulatory approval process ensures the development of safe and effective assays and medical devices. In this section of my talk, I'll focus mostly on the specifications of the FDA guidelines required for molecular assays validation. However, the development principles for both molecular and immunological assays are the same. Some of these main development principles for both types of assays entail limit of detection, analytical sensitivity, cross-reactivity, analytical specificity, microbial interference, and a clinical agreement/evaluation study. I will break down each method and expand on the molecular assay specifications. For the limit of detection, RNA or inactivated virus can be spiked into an artificial or real clinical matrix, such as bronchoalveolar lavage fluid or sputum. Developers should test a dilution series of 3 replicates per concentration with the inactivated virus on the actual patient specimen, and then confirm the final concentration with 20 replicates. Limit of detection is defined as the lowest concentration at which 19 out of the 20 replicates are positive. To confirm the specificity of the analyte, so in this case SARS-CoV-2, cross-reactivity is evaluated with inclusivity-exclusivity studies. To evaluate cross-reactivity with exclusivity testing, there are 2 ways that the FDA recommends testing: wet testing, or performing an in silico analysis of an assay compared to common respiratory flora and other viral pathogens. Clinical evaluations are mainly used to confirm the performance characteristics of the assays, which include the sensitivity and specificity of using clinical specimens with or without SARS-CoV-2 infection. This is except for serological tests, in which the clinical accuracy should be established on human specimens from patients with microbiologically confirmed COVID-19 infection. To meet the expedited need for assay and research materials, ATCC has developed a diverse SARS-CoV-2 strain selection. As shown in the upper left table, this list includes the Washington, Hong Kong, Italy, and Germany strains. Genomic strains are downgraded from BSL-3 to BSL-2, and the heat inactivated strain is downgraded to BSL-1. This significantly decreases the time and equipment required for research. However, with this in mind, countries out of the US may not follow this convention. These strains can be used for a variety of applications, such as limit of detection, sensitivity studies, clinical studies, and as positive controls for PCR-based assays. Quantified controls can also be used to monitor run-to-run variation within each step of required procedures, such as nucleic acid extraction, process verification, or amplification. The Washington strain, which serves as a SARS-CoV-2 reference material for the US, and the Hong Kong strain are also fully sequenced, assembled, annotated, and validated using our standardized workflow. The Italy strain is currently being sequenced and will be provided soon. All of these sequenced strains are provided on the ATCC Genome Portal. ATCC also provides 3 synthetic molecular standards, with 1 more in development, for use as controls in the development of molecular assays designed to detect and quantify SARS-CoV-2. These standards are synthetically derived and eliminate the need to culture viruses in the laboratory, and are safe to use under BSL-1 conditions. The standards have been designed to contain fragments representing important regions of the SARS-CoV-2 genome and to be compatible with the CDC and World Health Organization's RT-PCR assays. Construct 1 was designed to contain portions of ORF1ab, nucleocapsid, envelope, RDRP, and NSP14 genes, and are also designed to be compatible with the CDC RT-PCR diagnostic panel, and also the World Health Organization recommended real-time RT-PCR assays targeting the previously mentioned genes. While constructs 2 and 3 contains portions of the 5' N and 3' N of the spike protein respectively, and is compatible with the assays from the National Institute of Infectious Diseases in Japan from the WHO assay recommendations. Construct 4, which is still in development, contains portions of NSP9 and NSP12, and is compatible with the assays from Berlin, Germany, and Institut Pasteur. Overall, ATCC synthetics are artificially constructed RNA standards, which are precisely quantified by qPCR and ddPCR to ensure a precise copy number, as they are ideal for use as positive controls for PCR-based assays, and those requiring a standard curve in order to determine viral load. Other applications include limit of detection, sensitivity studies, clinical studies, and can be used to monitor run-to-run variation within each step of required procedures, such as nucleic acid extraction or amplification. ATCC synthetics has been also outlined on the FDA's website on SARS-CoV-2 resources on the positive control materials. To support the need for exclusivity testing material, ATCC provides a wide spectrum of microbial strains that are geographically and temporally diverse. This includes a diverse selection of respiratory flora and viruses, which are recommended for regulatory approval by showing sensitivity and exclusivity during assay development. In addition to diagnostics, the scientific community has developed novel vaccine strategies and experimental antivirals to fight this pandemic. Currently, there is no approved human coronavirus vaccine, since new vaccines specifically targeting SARS-CoV-2 are under development, and many new technologies used for production platforms or vectors are not yet tested for safety. Vaccine platforms that have been developed include RNA-based, DNA-based, recombinant protein-based, vectored, inactivated, and live attenuated vaccines. To develop new vaccines, researchers need access to virus isolates, and during the outbreak of a novel virus like SARS-CoV-2, it can be challenging to determine which propagation host is ideal for successful viral replication. In a recent study by Harcourt et al., it was discovered that SARS-CoV-2 could replicate to a high titer in Vero CCL81 and Vero E6 cells in the absence of trypsin. These cell lines and the media needed to cultivate them are available at ATCC. ATCC also used cutting-edge CRISPR-Cas9 gene editing technology to develop STAT1 knockout cell lines capable of producing high-titer viral stocks. We have application data presented in a recent webinar showing that these knockout cells produce model clinical viruses such as dengue and influenza A at titers tenfold higher than the corresponding parental cell line. This has the potential to significantly reduce cost associated with virus and viral vaccine production. Current vaccine research is extremely promising, especially given that ongoing research on SARS-CoV-1 and MERS coronavirus have identified a spike protein to be ideal vaccine targets for coronaviruses. In SARS-CoV-1 and SARS-CoV-2, the spike protein interacts with the receptor ACE2, and antibodies targeting the spike can interfere with this binding, thereby neutralizing the virus. All of this offers important insights and guidance to therapeutic antibody and prophylactic vaccine development. To expand on therapeutics, the host dependencies of SARS-CoV-2 are constantly being explored to identify other host proteins already targeted by existing drugs. Unfortunately, limited knowledge of the molecular details of infection precludes evaluation of some candidates for host-directed therapies. So far, no clinically available antiviral drugs have been approved, but clinical trials are ongoing. Screening how coronavirus proteins interact with human proteins to determine potential druggable sites involves existing drugs or compounds, live virus, and the cells in which to test them. Determining which cell line to be used can be challenging to find successful viral replication to test and develop antiviral compounds. To address this challenge, ATCC offers CCL81 and the Vero E6 cells an appropriate medium needed for cultivation. As mentioned before, SARS-CoV-2 can replicate to a high titer in these cells in the absence of trypsin. ATCC also offers the STAT1 knockout cell lines capable of producing high titer viral stocks, which has the potential to significantly reduce costs associated with virus and viral vaccine production. The host interactions of SARS-CoV-2 are being explored to evaluate and determine drug targets for antiviral development. Drug targets include remdesivir, lopinavir, epitene, and so much more. With current research, ATCC has been shown to play a prominent role in testing, as their materials, specifically cell lines in this case, are used heavily for virus-host-drug interaction studies. Along with the progress in determining new drug targets for antiviral development, there has been promising research conducted by multiple institutions, such as Mount Sinai Hospital and Institut Pasteur, in establishing whether existing drugs can be used to treat infection to expedite treatment, with the idea that the existing are at least safe. On the left side is a map detailing the possible inhibition pathways of various potential antiviral drug targets, indicated by the orange dots. These hinder critical functions of the virus. So every place that a coronavirus protein interacts with a human protein is a potential druggable site. Some of the potential drug targets include the mitochondrial ribosomal proteins, MRPS27, S5, S25, and S2. All of the proteins involved in quality control networks for the endoplasmic reticulum to optimize proper folding and trafficking of covalent proteins, and also the centrosome-associated protein CEP250, which plays an important role in centrosome cohesion during interphase. The scientific consortium identified two sets of pharmacological agents that displayed antiviral activity, such as disrupting translation of the virus and sigma receptors. In these efforts, ATCC products continue to be shown as a reliable source of various cell lines, specifically different virocells in antiviral research. Given the high number of infected patients and at-risk individuals, the current situation provides an opportunity to initiate clinical trials for vaccine formulation designed to protect uninfected people, broad-spectrum antivirals to treat infected individuals, and formulation of immune modulators to alleviate clinical pathologies. There are ongoing clinical trials with 341,642 studies in total being conducted all around the world to expedite the need for therapeutics, which makes the availability of treatments extremely promising. In summary, COVID-19 remains a high global and public health threat, and the development of safe and effective diagnostics, prophylactics, and therapeutics will depend on credible scientific research. And this much needed credible scientific research depends on credible biological material, which ATCC provides to expedite research and development, as we are committed to providing the high quality reagents to the global scientific community. With the continuing progress in each of these research fields, it is assuring that this pandemic will be under control and in decline, as we are all in this together to limit the spread of infection. Thank you to the BSL-3, Bioproduction, MSAT, and other ATCC teams who have greatly contributed to these efforts. And thank you to everyone for joining and listening.
Well, thank you, Brittany. In just a few moments, we will begin our Q&A session. Please use the chat function available through the webinar program to submit your questions. The recorded webinar presentation will be available on demand on the ATCC website at www.atcc.org. All right, so our first question is around vaccine development tools, specifically the CRISPR edited cell lines for vaccine development that ATCC provides. Can the STAT1 knockout viro cell lines produce more coronavirus than, I guess that would be the parental viro cell line?
While we know that SARS-CoV-2 will grow in the STAT1 knockout cells, they have not been tested yet for efficacy with SARS-CoV-2. So some of the groups are looking into this, but we do not have an answer yet.
Okay. Sort of changing topics here. Why are bats seemingly more susceptible for origination of the next pandemic?
Bats are more susceptible because they are a natural reservoir of coronaviruses and other deadly diseases, because it has been hypothesized that their immune systems have evolved to tolerate viral infections. Coupled with the fact that they are the only flying mammals, viruses can be transmitted very easily.
Okay. Now, the next couple questions we've got coming in are around coronavirus tests and diagnostics. Here's the first one. In the US, how many molecular-based and serology-based tests have received emergency use authorization?
Great question. As of June 8th, there are 91 authorized molecular and serological tests in total.
Okay, good. So that sounds like there's a lot of people working on this. Here's another one around the diagnostic tests. What is the range of LOD? That's a limit of detection, right?
That is correct.
Achieved by the molecular-based test.
Generally, the limit of detection is pretty low, but there is a wide range of measurements because reported genome copies vary greatly in different commercial kits.
Okay. So you'll see kit-to-kit differences, I guess, in the LOD.
Right.
Now, okay. This next question we have is around risk factors. Why is obesity such a high risk factor for severe cases of COVID-19?
This is because obesity increases the risk for breathing problems, such as ARDS, which means patients may not get enough respiratory support if hospitalized. It can also increase risk for other underlying conditions, such as making it more difficult for the patient to fight off the infection.
Okay. All right, we've got a couple questions coming in around vaccines against SARS-CoV-2. So, how effective could a vaccine be to prevent COVID-19?
While some vaccines do offer long-lasting protection, such as the polio vaccine, some only offer a short-term protection like the seasonal flu. While the SARS-CoV-2 virus has not shown much mutation across all the genotypic strains evident during COVID-19, it is still unknown whether it will make an ideal vaccine candidate. So there is really no way to predict how effective or long-lasting a novel coronavirus vaccine would be.
Okay, yeah, that makes sense. Now, are there any vaccines or other medical products to prevent COVID-19?
At this time, there is no vaccine to prevent coronavirus disease 2019. Vaccine developers and other researchers and manufacturers are expediting the development and availability of different medical products such as vaccines, antibodies, and drugs to prevent infection.
Okay, we've got another question around vaccines here. Who is making vaccines to prevent COVID-19?
Dozens of research teams from around the world are working to develop and test coronavirus vaccines. As of early June, the World Health Organization stated that there were more than 120 candidate vaccines under preparation.
All right. Well, at this time, we will conclude our Q&A session. I'd like to thank Brittany for the excellent presentation, and thank you everyone for attending the webinar. One final word. On June 25th, bioinformatician Tasha Santiago Rodriguez will present her talk entitled Viral Metagenomics and the Use of Standards, from Biology to Clinical Applications. Thank you again for your time today, and have a great day.